Thank you so much for inviting me to speak here, and I'm also really happy with Raghavan's talk and Erica's talk that I think set up this area of protein stability and really high throughput measurements in a great way. So the question, oh, they said it was going to appear over here. The question that I'm going to talk about is how does protein sequence determine protein folding stability, and then how do we predict protein folding stability? And unfortunately, I'm not going to get all the way to prediction, but I hope you'll be able to see how the experimental scale that we're doing, I think, will really improve that. So first, I guess, a lot of people study function in this room and are really excited about these high throughput assays for function. I'm focused on stability, and I just wanted to give a quick introduction of why we should care about stability. Well, first, everyone knows that folding stability is really important for function. Of course, it's not the only thing, but it's one important piece. And also, if we can understand stability, then we can learn about one particular mechanism by which function can go wrong. And being able to determine these two things independently gives us a better picture of like what causes a functional change. Stability, of course, strongly shapes fitness landscapes and influences how proteins evolve. Designing highly stable proteins is actually a real bottleneck right now, and improved models of stability would accelerate protein design. And lastly, protein stability is determined by the same biophysical forces that influence lots of other biophysical properties like conformational change, protein-protein binding, protein-ligand interactions. And a lot of these things are more difficult to measure on a large scale than protein stability. So if we can kind of understand these forces better through experiments and modeling on protein stability, I really hope that transfers over to improved understanding and modeling of these other areas of biophysics. Now, protein stability understanding has basically been approached in two different ways, through physical modeling and machine learning. But both of these have had real challenges that have prevented us from developing really accurate models of stability. So I'm going to quickly go through these. On the physical modeling side, so one of the big challenges of understanding protein stability is that stability is a small difference between two sets of really large numbers. So you've got adding up all the interactions within the protein on one side, and you've got this huge loss of conformational entropy that happens when the protein folds up into one shape. And the amount of energy kind of on both of these sides is much larger than this small difference that determines whether the protein's stable or not. Second thing is that each single interaction in the protein, like a hydrogen bond, can basically be as strong as the entire global stability of that protein. So you get a few interactions wrong, and you can predict the entire protein stability incorrectly. Another thing is that just the types of biophysical interactions that lead to stability are really subtle and just have proven really difficult to capture in computational models. So things like solvation, electrostatics, conformational entropy, these are all, as in physics, we like to model things as like the harmonic springs or something. And these are actually just really challenging interactions to model in a simplified physical way. And then the last thing is, to determine kind of the overall stability of a protein, you want to consider its entire conformational space in the folded state. Or if you want to consider the effect of a mutation on a protein, you really need to understand how that mutation is going to affect the entire conformational ensemble. And this kind of conformational sampling has always been really difficult. So now, jumping over to the machine learning side and why protein stability has been so hard to understand through machine learning, I think this can be summarized by the joke that the food is bad and the portions are small, that there's always been it's been difficult to measure stability, and there's been a limited amount of stability data that people could use to train machine learning models on. So historically, people have used databases that are collections of stability measurements like Protherm, but Protherm was never designed to be a comprehensive set of data for machine learning for protein stability. It's like one lab measured 10 stabilities over here, and a different lab measured 10 stabilities over there. And of course, stability depends on the experimental conditions that you use of temperature, pH, salt. And when everyone's collecting this in a kind of unorganized way, it ends up with a data set that's really not optimal for machine learning. We also have a heavily skewed set of proteins and mutations in protherm. So the vast majority of mutations that people have studied are mutations to alanine. They're very heavily weighted toward destabilizing mutations. And one of the, of course, main goals of understanding stability is to design more stable proteins and learn how to increase stability. And then finally, the goal of protein stability modeling for a really long time has just been to understand mutations, like the difference between one sequence and another, because when all the data has been collected under different experimental conditions, that's really the only thing we could try to put it all together to do, to understand effects of mutation. But what I hope the direction that this field can go in is to actually predict the absolute stability of protein domains under particular temperature and experimental conditions. And I think this will ultimately prove more powerful and will probably ultimately be a better way to predict differences of stability caused by mutations. Okay, so the strategy that our lab is taking to try to understand stability is to develop new, really large-scale methods for measuring folding stability. And the work that I'm going to tell you about was done by a postdoc in our lab, Kotaro Sugiyama, who actually arrived just before the pandemic started, went home for a few months, and then was back in the lab working basically by himself. And now he just started a junior PI position at University of Tokyo a few months ago, and we really miss him. Okay, so the approach that he developed is based on this technology called cDNA display, and we didn't develop it, but basically cDNA display is a fully cell-free method where we put in DNA and we use DNA oligo pools, and then those get transcribed and translated in the cell-free mixture, and what we end up with is proteins that are covalently linked at the C-terminus to the cDNA. So initially, they're transcribed from the mRNA, but then we reverse transcribe that mRNA into cDNA, and it's actually the cDNA that gets covalently attached to the protein, and it's like a little bit more robust version of mRNA display, if you're familiar with that. So we work with DNA oligo pools, and what that enables us to do is encode each different domain that we want to look at on an individual oligo, and then customize all the different proteins that we put into an experiment. So the downside of this is that up till now, our experiments have been really limited to small domains that we can encode on individual oligos. But I still think that these small domains are going to be really useful training data for machine learning. So the largest experiments that we've done with this method have been 900,000 different protein sequences in an experiment. And what we usually find is that we get really good, if we pick our sequences well, we get really good data on pretty much every sequence that we put into the experiment. So this is the cDNA display method that I just told you about. We have the proteins covalently attached to the cDNA on the C-terminus, and then at the N-terminus is an epitope tag that can get pulled down by an antibody. And so we take this protein mixture and we incubate it with proteases, and at least for these small domains, the small domains basically have to globally unfold in order to be cleavable by the proteases. And we use trypsin and we use chymotrypsin that cut in different places. So trypsin cuts after positive amino acids, and chymotrypsin cuts after aromatic amino acids. So these amino acids are found in basically all domains. We don't have to program in specific cut sites. But by doing separate experiments with these two separate proteases, we can validate that our results are not specific to just one protease, that we're seeing the general folding stability rather than something that's determined by the protease that we're using. And then after incubating these with protease, the last step is to pull down the intact proteins by the N-terminal tag. And as long as the protein's still intact, it's going to carry down the cDNA, which we can then sequence. So we do this at a range of different protease concentrations. I'm showing 12 or something here. Our actual experiments, we use about 24 protease concentrations. And so everything starts out the same amount in the library. And then a very unstable sequence, like this dark blue, becomes depleted in the library once we get to the protease concentrations that will cut it up. And when this is getting depleted, other stuff is getting enriched. And each sequence starts to get depleted at some higher and higher protease concentration. And so this red sequence up here is one of the most stable sequences. This is the wild type of this Fin SH3 domain. But it's still actually not getting enriched as fast as the fastest stuff, which I guess are stabilized variants of this domain. So from all this, we put all this data into a model that actually helps us infer these cleavage curves for each different protein in the library. And what we extract from that is this K50 value, which is not quite the protease concentration that leads to 50% cleavage. It's protease concentration that leads to 50% of the maximal cleavage rate. But anyway, this K50 basically captures how protease sensitive is that domain. So then I told you we want folding stabilities and not just protease sensitivities. So to convert these protease sensitivities into actual like folding stabilities, delta Gs, we built another model that basically predicts how fast is a sequence going to get cleaved in its unfolded state. And so how fast a sequence gets cleaved in its unfolded state depends on how many different cut sites for that protease does that sequence have. Well, now we just switch screens. The mouse is moving. So anyway, hopefully this will come back shortly. But we compare the experimentally measured protease stability of a sequence to what we predict that sequence would have in the unfolded state. And we measured a bunch of unfolded stuff in order to calibrate this model. And then the difference between those two gives us the folding stability. All right, we're back. The difference between those two gives us the folding stability of all the sequences that we're looking at. So these are our folding stabilities calculated with one set of experiments using trypsin. And these are our folding stabilities with a different set of experiments calculated using chymotrypsin. And you can see that at least for a good chunk of our data, we're showing the data that we picked out here in part based on this agreement, but in part based on other factors, showing that these two independent experiments lead to consistent folding stabilities for these are like our highest quality set of data, about 770,000 different protein domains. So, does this work? Like, how does this compare to measuring one protein using a traditional biophysical assay? So what I'm showing here are data from Alex Nisthal, who, when working in Steve Mayo's lab, designed a robotic system to individually purify each different mutant of protein GB1 and do kind of a traditional chemical denaturation assay. And so these are their stabilities measured one at a time, and these are our stabilities measured using this proteolysis approach. And you can see that they're very consistent with each other, with some outliers that we could talk about in the questions if you're interested. So that's great. And the data that we collected here for all the mutants of this protein G domain, we didn't do just this domain by itself. These data were actually collected in a library of 700,000 different sequences. So this is the quality of agreement that we get to individual measurements, even when doing this on a really large scale. So that was protein G. And here I'm showing you essentially those same plots for a bunch of mutants of 10 different domains. So there are definitely limitations here. All these domains are small. They're all shorter than 80 amino acids, but they cover a range of different fold types, from like all alpha domains to an SH3 domain that's all beta, WW domains, the mixed alpha beta of protein G. And in each of these different cases, we see a really good correlation between our high throughput folding stabilities and individual measurements that, from purified proteins that have been published in the literature over the last 30 years. So what did we do with this approach? Well, the main thing that we've done so far is collect tons of deep mutational scanning data. So this is all the point mutants of a human E4B ubiquitin ligase. And along with all the substitutions, we also did 2 insertions at each site and deletion of that site. And for me, this is really cool because if you look at databases of stability, there's hardly any insertions and deletions. So it's nice that by using oligo pools, we just order the sequences that we want, and we were able to characterize these as well. This is the N-terminal domain of the E. coli arginine repressor. These are some more mutational scans. These are some more mutational scans. I feel like I actually need one more slide because this audience isn't going to be impressed by just this amount of data. So I really needed to go out to one more zoom, but that's as far as I went. OK, so I showed you that this really works a lot of the time, but there really are limitations to this method that we want to improve in kind of our next work. So like I said, most of the time we see really consistent measurements between trypsin and chymotrypsin. And so I'm showing two examples of well-behaved domains. This is what some of our data looked like, where we actually saw real differences between trypsin and chymotrypsin. So the chymotrypsin data on the y-axis for all the mutants of this domain are spread out over about 4 kcal per mole. The trypsin data only goes up to about 2 kcal per mole. So basically, everything's getting cut by trypsin before it's getting cut by chymotrypsin. And if we look at this particular domain, there's this really big loop that happens to have 2 arginines, which are trypsin cut sites. So when we have domains that can get cleaved without having to globally unfold, then our assay is not going to work. And this is one of the important things that we need to improve kind of in the next generation. And we can actually confirm this by looking at the mutational scanning data with trypsin. And this is one of those arginines where you can see that basically mutating away this cut site or the other cut site next to it really stabilizes the protein. And of course, it's not actually changing the folding stability. It's just removing this arginine cutting site. And if we remake this wild type that no longer has this cut site, then now, I'm not showing the data, but now we get one of these really consistent correlations between trypsin and chymotrypsin because now they're both properly measuring global folding stability. One of the other challenges that we ran into was the limited dynamic range of our assay. So I showed you that after we do this proteolysis, we pull down all the domains by their N-terminal tag. And when we go up to really high protease concentrations, that tag is one thing that can get cleaved, or maybe there's other parts of our fusion construct that can get cleaved. But basically, we kind of have a maximum effective range with at least this iteration of the assay. So these are all the point mutants of ubiquitin. And pretty much everything is white with a slash through it, which means it was at the top of our dynamic range, and we couldn't resolve any of the stabilities of these mutants. But one of the cool things that we did in this paper was when we saw proteins like this, we made destabilizing mutations and then remeasured all the single mutants in that destabilized background. So this is all the point mutants of ubiquitin in an I3A background, and this is all the point mutants of ubiquitin in a L67S background. So here's A3, and you can see that it's slightly stabilizing, mutating it back to I, although I think you can't see the full extent of that stability because that actually brings it outside the range of the assay. So we have a bunch of data like this of all these mutants in destabilized backgrounds, which we haven't really investigated that much yet, but it makes it possible to investigate epistatic effects of are mutants behaving differently in this background compared to this background. In this particular case, there's a really good correlation between what we see in these 2 different destabilized backgrounds. So it lets us, even though the assay has this upper dynamic range limit, it let us still measure the effects of mutations in these really highly stable proteins. So what have we done and where are we going? We've collected a ton of stability data with this method. We've measured about 3 million stabilities to date, and they're all under identical experimental conditions. And when we measure the same sequence in 2 different libraries, we get consistent results across those different experiments on the same sequences. We've done deep mutational scanning of 354 natural domains and 188 design domains, again, that are all kind of smaller than 72 amino acids. We also collected about 200,000 double mutants. And this is not like the double mutant data that I just showed you on ubiquitin. We took 600 pairs of sites where 2 amino acids were interacting and made all the double mutants like 20 by 20 at those sites to basically learn how different amino acids interact. And I think that's really useful data for machine learning. And we've also measured about 300,000 just very diverse domain sequences from metagenomics. So not deep mutational scanning, but just all the different types of folds of domains that we could find in nature. And I think, again, that's going to be really interesting for machine learning. So you can read about this. There's a preprint on bioRxiv, megascale experimental analysis. I thought the name megascale was cool, but like now whenever people talk to me, they're like, there's even other papers that say we use the megascale data set for this, so the name kind of caught on. Yeah, it has about 770,000 single and double mutant stabilities. And so not only is it on bioRxiv, all the data is downloadable from Zenodo, and thanks to Alan, it's also in MaveDB. So like I said, there's a lot of things that we need to improve for this method. The dynamic range of the assay is currently limited to about 0 to 5 kcal per mole, which is actually basically covering the range of most human domains, but we'd still like to be able to extend that to measure to resolve really stable proteins. And I think one of the ways we'll do that is by using denaturant in these assays instead of a range of protease concentrations. We want to expand the data set to larger proteins, especially taking advantage of oligo pools that can now encode larger proteins, or ways of linking oligos together to still make really diverse libraries of larger proteins. Also, something that I've thought about a little bit is just trying to collect all the DNA that's already been made for different MAVE libraries. But I think our assay is going to be most relevant to single domain proteins, because if your protein has a bunch of different domains, we're basically just going to resolve the stability of the least stable domain rather than kind of each different domain together. But yeah, how we should get DNA is an interesting topic, and I'd like to hear people's perspectives on that. Yeah, we're still limited in the types of domains that we can look at. If the domain has disulfide cross-links, then the DNA is not going to dissociate after protease. We can probably improve the assay in this direction. And then when we picked all these domains, we basically just picked everything that was in the protein data bank under these size limits and didn't think at all about the disease relevance of different domains. So if there are domains that are important for health, amenable to the assay, and where a stability measurement would actually be really valuable as opposed to a functional measurement, then that would be really great, and we should put all that into our experiments. The long-term goal for me of collecting all this data is to build a predictive model, and I think like an AlphaFold style model for stability is really believable. I said at the beginning that I think that the focus of stability prediction should be predicting absolute stabilities of domains rather than just the effects of mutations. And that's because the effect that a mutation has depends on the overall energy landscape of a protein. It's not just what amino acids is it touching, because it's how does the entire protein react when you make that substitution. And I think having all this data under identical conditions of absolute stability should really improve predicting the effects of mutations. The absolute stability of a protein is also kind of what's most relevant to different phenotypes. If you change a protein from 9 kcal per mole to 7 kcal per mole, that's going to have maybe a smaller phenotypic effect than 2 to 0 or 1 to -1. Absolute stability is also most relevant for protein design. I think transfer learning from models that are trained to predict protein structure and then building stability prediction on top of structure prediction is going to be incredibly powerful. And to get there, what we're doing is just trying to measure stability for this huge range of diverse domains. So I just want to acknowledge my lab, especially Kotaro Tsubayama, who did this whole project, other people in the lab who really made it a great environment to work in, different people who contributed to different aspects of the paper that I didn't talk about in this talk, but still all their contributions were really great. And then funding for this project mainly came from postdoc fellowships from JSPS and Human Frontiers program to Kotaro, and the EME Corporation for providing the cDNA display reagents. So thanks, and I'm happy for your questions.
Looks like we've got an immediate question over there in the back.
Hi, thanks for a great talk. Does it make sense to use this approach to look at intrinsically disordered regions of proteins, or does that not translate here?
If you have an intrinsically disordered protein, so our experiment doesn't really tell you where a protein gets cut. It just tells you how much protease do you add or how much denaturant do you add before you get cut. So if you have intrinsically disordered regions, then you're just going to see that is a unstable protein. And so I think this is maybe not interesting in the way you...
Got it. There wouldn't be as much signal as I would want.
Yeah.
Great. Thank you.
Benny?
So following a bit on this question, is there anything in the accessibility to proteases or in the overall mutational signature that allows you to distinguish an aggregation-prone region from a super stable one?
One of the problems with our assay is that aggregation-prone proteins look like they're folded and stable. And I think there are assay improvements that we can try to make to get around that, like maybe include small amounts of detergent in the experiment. When we do mutational scanning, we see that well-folded domains have mutational scanning heat maps that look like what we're used to looking at. When we look at what we see a different class where basically the whole protein is not sensitive to mutations, or putting new hydrophobic amino acids anywhere in that protein appears stabilizing. And we interpret those signatures as indicating aggregation, but I couldn't prove, we haven't shown that that's actually what's happening.
Okay.
Here.
There's a question down here, Matt. Oh.
Hello?
Oh, up there, sorry.
Yeah. Can you do that to screen for ligand binding in a scenario where you add a ligand and they will change the stability of your protein domain?
Yes, we haven't done that, but it should totally work. And you could screen across all these different domains. You'd still have to do a separate experiment for each different ligand, unless we somehow connected like a DNA barcoded ligand to this or something. But I think that would be cool to do.
Matt?
It's a lovely talk, and kudos to you and Alan for getting it into MaveDB.
Alan, yeah.
So I was gonna. So do you have the data that might allow you to explore species differences in protein stability? And wondering if you look and domains that sequences that come from thermophilic organisms.
Yeah. So in the data that we've put out so far on all this mutational scanning, we have stuff that crosses a bunch of different organisms. We haven't, since it's mutational scanning on only a few hundred domains, it's probably not exactly what we want for that. On the metagenomic data, well, that might be interesting, except we don't know what species all that stuff came from. But that's more maybe the type of data that we would want.
We'll go over here, and then Debbie.
Hi. Membrane proteins represent a third of the human genome. Do you have any plans for adapting the assay so you can actually consider the folding in the membrane?
Yeah, so my colleague Anastasia Vorbieva in Belgium is basically trying to do cDNA display in a large-scale screen with membrane proteins, and I hope it works. It won't be like GPCRs right away or something, but I hope that this will be relevant to membranes, and she's trying it right now.
Okay, last question, Debbie.
Great work, Gabe. And actually following on from Matt's question, he keeps doing this to me. And thinking about the thermophilic organisms, I mean, it'd be interesting to see, maybe you've already looked to see whether your stabilizing mutations are actually ones that correspond to those corresponding domains in thermophilic organisms. I mean, that's the trick that crystallographers have used.
Like if stabilizing mutations that we find
are actually the same as the amino acid that are used.
Yeah. That would be interesting to look at, and we haven't looked at it at all.
Okay. Thank you for all these awesome questions. Thank you, Gabe.
Thank you.